Today we're going to be talking about bacteria and archaea. So we are done talking about evolution and how it occurs, how that change in allele frequency over time occurs, and now we're going to talk about diversity of organisms. And does anybody know what this organism is? It's a squid. You know it's not a cuttlefish because cuttlefish, their eyes have a W shape in them. It's an easy way to distinguish them. This is actually a Hawaiian bobtail squid. And no, it is not a bacteria or an archaea. It is a eukaryote, which we will be talking about on Thursday. But this bobtail squid looks relatively normal, but when it is in darkness, it actually bioluminesces, it lights up. And the way that it's able to do that is with a symbiotic bacteria. And this symbiotic bacteria gets a safe place to live and easy access to food by living in the squid. And the bioluminescence, that light that the bacteria produces, actually helps the squid hide from predators when it's deep down in the ocean. Predators swimming underneath it, look up, and the light that this bacteria makes actually mimics the light that's coming down from above, from the sun. And so these squid actually blend in to their surroundings so predators cannot see them. So just a really cool example of a cool, good bacteria. So bacteria and archaea are very diverse groups of organisms. These are microscopic organisms, we cannot see them, and they're very diverse in their shape, in where they're found on the planet, but they are more numerous than all of the eukaryotes put together. One of the reasons we didn't know this until the past few decades, is because most of these we cannot culture in a laboratory. Less than 1% of bacteria and archaea are able to be grown in a lab. So we have to be able to somehow test these in the environment. And now we can use molecular tools to do that. So bacteria and archaea are part of the group called prokaryotes. Prokaryotes are all unicellular, so they are single-celled. If they're not single-celled, it's because they're just living in little groups. Their cell size varies tremendously, but most of them are less than 1 millimeter in diameter. So these are very small, most of the time microscopic organisms. They have no membrane-bound organelles. And their DNA is not bound by a membrane, meaning it is not in a nucleus. So here we have a picture of a bacterial cell. You can see the DNA, which is in a ring. And they have ribosomes, which help to which actually make the proteins in the cell. But other than that, there's no organelles, there's no internal membranes. There's just the outer cell wall. That is their membrane. So if we're looking at relationships, here's a phylogenetic tree, and we have our most recent common ancestor of all living organisms, and it had a prokaryotic cell organization. So the DNA was not found in a nucleus, there are no organelles, it's a single-celled organism. A lot of you were asking last week, what is the ancestor to bacteria? We don't know, because there's no fossil record of these organisms. Later in class, we will talk about one of the most ancestral bacteria that's still alive. You can see bacteria is here, here's archaea, and then on Thursday, we will start talking about eukaryotic cells and organisms, which have a very different type of setup compared to the prokaryotes. So your first clicker question of the day. Wow, that happened fast in class. So, prokaryotes, both bacteria and archaea have certain things that are in common that we can talk about that distinguish them from eukaryotes. The first is that cellular division typically occurs through binary fission. In binary fission, you have your single-celled organism, the DNA replicates itself, and then the cell membrane splits and you end up with two identical daughter cells. So in binary fission, the two offspring, the two daughter cells, are identical to the single parent cell. They do not reproduce sexually like we think of eukaryotes doing. So you might be wondering, well, how do we have so much variation in the different types of bacteria and maybe archaea, if you know about them? How do we have variation in our genes? Because other than mutations, there's no way, if they're not reproducing sexually, for these organisms to transfer their genetic material. What these organisms do is they use horizontal gene transfer. So this is the exchange of genetic material only by sharing. So they're not reproducing, they're just sharing. They're transferring their genes. So we can have a donor cell, and in both cases, there is a recipient cell that will take up the DNA. There are three different ways that these types of cells can do this. The first is conjugation. So conjugation, we have our donor cell, they make a little pilus, and the DNA can go across to the recipient cell. So literally it's like they make a little hallway and they share some DNA. The second way is by transformation. And in transformation, what happens is a bacteria cell, for example, will die. As its cell membrane breaks down, that DNA is released into the environment. And then a different cell can pick up some of that DNA that's just floating around in the environment. So that's transformation. And the last is by transduction. And this occurs through a virus. So a virus infects the donor cell. It picks up some of that DNA from the donor cell. And as it infects a new cell, it also inserts that DNA from the original donor cell. We can use transduction in research to figure out what different genes in different types of organisms do. So people can actually do this in the lab for research purposes. So I said that prokaryotes are very small in size. Most of them are smaller than 1 millimeter. A lot of them are even smaller than that. So why do they have to be so small? The reason that they are so small is that they get nutrients through the process of diffusion. So this is a passive way for them to get nutrients. The nutrients come in passively through their cell membranes, and then those nutrients need to get to the rest of the cell. As a cell increases in size, it needs more nutrients, so more nutrients are going to need to diffuse into the cell. So you need a larger surface area, so the outside, think of the membrane of the cell, that's the surface area. You need a larger surface area to volume, so what's inside the cell. You need a larger surface area to volume ratio to ensure that the nutrients get to the interior of a cell. So here I have two very simple cells that I drew, and you can see it takes a lot more distance for my little green nutrient to get in to the center of the cell if it's bigger. As you get bigger, your volume increases faster than your surface area. So these single-celled organisms are constrained by their ability to take up nutrients. And down here is just a 3D version if you're trying to think about an actual cell that is three-dimensional instead of two-dimensional. So cell size is restrained due to nutrient uptake, which happens through diffusion. So all organisms need to acquire energy and they need to acquire carbon. They need to acquire the basic building block of all organic molecules. So if we're looking at this schematic, we have all organisms, because all organisms, whether you are an animal, a plant, a fungi, a bacteria, an archaea, a protist, you need to get energy and you need to get carbon. Some organisms get energy from the sun. You learned about photosynthesis last semester, if you took 150 last semester. Organisms that get energy from the sun are called phototrophs. There's two different ways that you can get carbon. You could get carbon from inorganic sources, such as carbon dioxide, or you could get carbon from organic compounds. If you get carbon from inorganic compounds, think of most of the plants that you learned about last semester, about photosynthesis. So those organisms are going to be photoautotrophs because they get energy from the sun, carbon from inorganic sources. So we're more familiar with vascular plants. An example from bacteria is cyanobacteria, which is able to take energy from the sun, get carbon from carbon dioxide, and survive. We can have photoheterotrophs, so you get energy from the sun, you get carbon from organic compounds, and an example from bacteria is heliobacteria, which are circular-shaped bacteria that can get energy from the sun, but they need to get carbon from organic compounds such as sugars, like glucose. And then most green bacteria also does this. If we're thinking about animals, we do not get energy from the sun. We have to get energy from chemical compounds. That's why we have to eat. So we are chemotrophs. We get our energy from chemical compounds. And just like phototrophs, chemotrophs can get carbon in two different ways. They could get it from inorganic sources or organic sources. So animals are chemoheterotrophs because we get our energy from chemical compounds and we get our carbon from organic compounds such as glucose. Sulfur oxidizing bacteria and hydrogen bacteria, so these are organisms living in low oxygenated environments, get their energy from chemical compounds, and then they get their carbon from inorganic sources, so they are chemoautotrophs. So this is just kind of a summary slide for you. Organisms have two sources of energy, the sun or chemical compounds. They have two sources of carbon, either inorganic molecules such as CO2 or glucose or other organic molecules, but typically it's glucose. So if you want to think about it more in a chart form, if you use light and CO2, you're a photoautotroph. If you use light and organic molecules, you're a photoheterotroph. If you use organic molecules for energy and CO2 for carbon, you're a chemoautotroph. And if you use organic molecules for both energy and carbon, you are a chemoheterotroph. Any questions? Great, because we have another clicker question. So now we're going to talk about the diversity of bacteria and the diversity of archaea for the rest of class. So bacteria diversity is great. There's a lot of diversity in bacteria. They do a lot of different things in the environment. Proteobacteria, which is this whole clade here, is the most diverse of all the groups of bacteria. So the majority of bacteria, most species, is found in proteobacteria. I apologize if you are red-green colorblind. This is what the textbook figure is. I don't know why they use red and green, but all of the green ones are all photosynthetic groups. So photosynthesis, the ability to get energy from the sun, evolved multiple times in bacteria. Some bacteria are good and they make us sick. We actually have some good bacteria, so good bacteria, bad bacteria, neutral bacteria, lots of different types of bacteria. Yeah. Everything in red are bacteria that they talked about in the textbook. Yep, they just wanted to let you know where they're at on the tree. Good question. So a really cool place where we can look at bacterial diversity are in microbial mats. And you can see that there's different colored areas in these mats. And the different colors are actually made by different types of bacteria that get their energy and carbon sources in different ways. So if you see this blue-green, this higher up layer, this is cyanobacteria. These organisms need oxygen-rich environments, but they can photosynthesize, so they can get energy from the sun. So they are found on the top of the surface because that's where they can get oxygen and sunlight. The next group, this purple color, are bacteria where they do not need oxygen, but they can still photosynthesize. So light is still entering into this area, but there's low oxygen, so they get their energy from the sun, carbon from inorganic sources. And then we have the big part on the bottom, this black navy blue color, and this is anaerobic respiration and fermentation. And so these are organisms that have low oxygen and no sunlight, so they have to get energy and carbon in other ways. And they live usually in the ground. So based on this picture, we have another clicker question. Switch, and we're going to talk about archaea diversity. And I think that these organisms are so cool. So these are, we don't know about these organisms quite as much as we know about bacteria because these were not discovered to be their own domain until 1977. And these organisms thrive in many different environments. Many of them are what are considered extremophiles. So they thrive in extreme environmental conditions. So the changing of colors on the branches of the trees is showing what their optimum growth temperature is. And you can see many of them can survive in temperatures of close to 100 degrees Celsius. So that's boiling water. Some have high salt tolerance, so they can live in very, very salty environments. And those are the medium brown colors, so you see some here and down here. Some are methane producers, so some live in the intestinal tract of animals such as cows that help them break down cellulose. They produce methane, and then cows release that methane. And some are even acid tolerance. They're able to live in very low pH environments. So these are just some pictures of different types of environments that archaea has found. This river is not supposed to be this orange color. This river is normally just regular water, but what happened is it became contaminated from coal mining. And so this river is now what is called acid mining drainage. So drainage coming from the mine has made this river very, very acidic, and the only organisms that can live there are extremophiles, which is including acid-tolerant archaea can live here, and that's one of the reasons why it's this rust color. Temperature-tolerant organisms in archaea can live in deep-sea thermal vents. They are actually the keystone species that allowed other organisms to survive in these deep sea environments where there is no sunlight. They also can live in geysers, such as these in Yellowstone. They are salt tolerant, and these are pools over in the San Francisco Bay. So when you fly to San Francisco in an airplane, you can look down as you're getting close to the airport and you can actually see these. And there are these really cool different colors. And what companies do is they have these ponds, and what happens is in this very salty water, these archaea basically take in the salt, and over time, companies can harvest salt for us to use from these pools. And the different colors are showing the different levels of saltiness. We also have archaea in our stomach and in other places in our body. So we actually have symbiotic archaea in our body. So some live in our gut, which is very acidic. They're able to tolerate that. So they're either symbiotic or they're just neutral. Most of you are probably wondering why I have this picture of this guy on here. This is Carl Woese. He was the person who discovered that archaea was its own domain. He discovered this in 1977 when he actually figured out a way to use molecular phylogenetics. So being able to look at the molecular basis of different organisms to help us determine how they're related to each other. So he is the father of archaea, and they say that his paper that came out shook the biological world the most since Darwin's Origin of the Species came out. So he's kind of a really big deal. If you want to learn more about him, let me know. I know all about him because he worked at the University of Illinois, and that's where I was for 6 years. So if we're looking at bacteria versus archaea, there are certain similarities between the two domains, and then there are some very distinct differences. So one thing that they both share in common is that their cells do not have a nucleus or other membrane-bound organelles. Their DNA does occur in a circular form, and it's free-floating within the cytoplasm. One difference they have is that their membraneless lipids are not ester-linked, and we'll talk more about that next class when we talk about how these organisms are related to eukaryotes. Same thing with their histone proteins. That's different. One thing that they have in common is they do not have introns present in their genes, so their genomes are much smaller. They don't have extra stuff. One thing that's really important to know about these two groups of organisms is that while bacteria is sensitive to antibiotics, archaea is not. So if you have an infection from an organism that is in the group archaea, you cannot take antibiotics because antibiotics target transcription and translation in bacteria. It's targeting these lipids and proteins, but archaea uses different methods to do transcription and translation. So they actually do not work. Bacterial antibiotics do not work on members in archaea. Any questions? Great, because guess what? Yay. Okay, so people were really curious about what's the common ancestor to bacteria and archaea and all living organisms, and we're not exactly sure. What we do know is we can look at bacteria, specifically these formations called stromatolites, to look at the fossil record. Stromatolites give us the oldest fossil records of life on Earth. So the way that this process occurs is that microbial mats form. So you can see that there's these mats. Sediment rains down, and then bacteria grow within the sediment. Then we have another microbial mat. Sediment rains down again. Bacteria forms. We have another microbial mat. And so if you cut these stromatolites, if you take a cross section, it's almost like looking at the rings on a tree. So you could see the evolutionary history. The further down you go, the lower into the ground the microbial mat is, the older it is. The further on the top it is, the newer it is. So we're going to watch a little video, and I actually embedded this video into here, so hopefully that works. Oh, nope.
Stromatolites are features of sedimentary rocks that form through the interaction of microbial communities and physical processes in lakes or shallow seas. The distinctive layered forms of stromatolites provide the most conspicuous record of life through the first 3 billion years of recorded Earth history. Stromatolite formation begins with the establishment of a microbial mat, a community of interwoven bacteria, usually cyanobacteria, on the sea floor. Sediment particles rain onto the mat-building microorganisms grow upward toward the light, trapping sediment particles and binding them into a layer. Within the mat, heterotrophic bacteria drive the precipitation of calcium carbonate minerals, which cement the layer in place. As the mat community forms a new surface, the process repeats again and again, resulting in the layered structure characteristic of stromatolites.
So you have access to this video because you can click on the link on the slides, and that's just an explanation of how stromatolites form. So there are benefits to prokaryotes. There are environmental benefits. They help chemicals cycle in the environment. They help with decomposition, so they help to break down dead or decaying organisms, which is great because if they didn't, they would pile up and we would have to deal with that, which would be gross. They help with nitrogen fixation in plants, so certain bacteria help take nitrogen from the soil, and then plants can use that nitrogen to grow, and then we get that nitrogen by eating plants. There's also certain symbioses that have formed between bacteria and archaea and organisms, such as in the digestive tract of animals. So these figures are from your textbook, and they were looking at the human microbiome. So humans and other animals have a microbiome that's made up of single-celled organisms. And what they did is they looked at the gut, so the stomach microbiome of African children who eat more of a plant-based diet compared to European children who eat more of a meat-based diet. And you can see that the diversity and the types of organisms that we find in our gut microbiome changes based on what we are eating. And so people who eat more plant-based diet have more organisms that help break down things such as cellulose. Without these organisms, we would not be able to get most of the nutrients out of the food that we eat. We can use things for genetic engineering, so bioremediation, so we can actually put prokaryotic organisms into the environment to help do things such as pollutant removal, so such as cleaning up oil spills. They will actually, some of those extremophiles will be able to break down the oil. We can use biofactories to make things such as vitamins and antibiotics. So most antibiotics we get from bacteria. And one that I think is really cool is we could use it in biological control. For example, we can take certain bacteria that's found in some insect species and implant it into mosquitoes that transmit diseases, and those mosquitoes can be released into the environment, spread that bacteria, and if that bacteria is in those mosquitoes, they actually are not able to transmit certain diseases. So we can use bacteria to help stop diseases that are spread by other organisms. And because I like mosquitoes and I think they're cool, this is actually a picture of mosquito cells with a bacteria called Wolbachia, which is in green, so you can see how it is infused in all of their cells. And when these mosquitoes have this Wolbachia bacteria in their cells, they are not able to transmit dengue virus. So that's an example of a